Hi friends, my name is Nandita. I am from Andhra Pradesh.
Now let us discuss the internal structure of Dicot-Root. The internal structure of a typical dicotyledon root showing the following features like epiderma, epiblema, cortex, endodermis. Epiblema. It is a uniseriated outermost layer. It comprises a tubular living components. Cuticle and stomata are absent. Unicellular root hairs are formed due to the elongation of some cells of epiblema. Epiblema is also known as rhizodermis and piliferous layer. Root hairs are present in maturation zone of root cells of epiblema, which is developed root hair called trichoblast. Cortex. Cortex is made up of parenchymatous cells. Chloroplast is absent, so they are non-photosynthetic, but chloroplast is present in roots of Tinospora and Trapa, so they are photosynthetic. The cells of outer part of the cortex are suberized in old roots. It is called exodermis, and exodermis is found in some dicotyledonous roots and most of the monocotyledonous roots are also. Endodermis. This layer is situated between the pericycle and cortex. Casparian strips are present on radial and tangential walls of endodermis. The strips are made up of lignosuberin, means mainly suberin. The cells of endodermis, which are situated in front of protoxylem cells, lack Casparian strips. These are called passage cells, transfusion cells, and the number of passage cells is equivalent to the protoplasm cells. Passage cells provide the path to absorb water from cortex to pericycle. Linearly arranged on root apex. The number of root hairs are similar to the number of passage cells. Casparian bands and passage cells are well developed in monocot roots. These are also present in stem, but they are less developed in endodermis, act as a watertight jacket. Pericycle. It is a single layer and composed of prosenchyma, a type of parenchyma. Pericycle of roots are single layered. At the time of secondary growth, pericycle is responsible for the development of crop cambium. Lateral roots are originated from the part of pericycle which is laying opposite to protoxylem. Those lateral roots are endogenous in origin. A few matured cells of pericycle, usually opposite to protoxylem, becomes meristematic. These cells divided by periclinal divisions and form some layers of cells. These divisions are followed by anticlinal divisions forming a perimordium which grows to form a lateral root. The branches of stems are exogenous in origin because this originated from the outer part of the cortex. The adventitious roots are also endogenous. It originates from phloem, parenchyma, or internal layer of endodermis or pericycle. Vascular bundles. Vascular bundles are radial and X-arch. Xylem and phloem are separated by equal in number. The number of xylem bundles are 2 to 6, but exceptionally, ficus means banyan tree, root is polyarch. Parenchyma, which is found between xylem and phloem, is called conjunctive tissue. Now let us discuss about internal structure of orchid root. These are the following features of orchid root. Velamen. Velamen, these are found in aerial or hanging roots of some epiphytes, like example orchid. These are the examples of multi-layered epidermis, and these are present outside of the exodermis. These are made up of protoderm and are dead. They absorb atmospheric moisture by imbibition. Passage cells are found in both exodermis and endodermis in hanging roots of orchids.